In this lecture we're going to go through the very useful tool analysis of variance. So what is analysis of variance? So we have an example here where we say, well, we know that food boosts your metabolism, and we want to investigate this, so we give a bunch of rats a normal diet and another bunch of rats a high-fat diet, and we wish to compare how they develop their weight over time. Eventually we want to include more than just two groups, so for instance saying a normal group of a normal diet group and then a high-fat diet with one type of protein and a high-fat diet with another type of protein, and we wish to compare all those three groups simultaneously. So that is the setup. So here is another example. We wish to compare the amount of ethyl acetate in wine produced at 4 different countries. And we see here a plot of the data that we have, and it seems so that Australia and South Africa have slightly higher levels compared to Argentina and Chile. So what is important in the comparisons of the differences between these different groups? Which statistical numbers should be used in this calculation? So please pause the video now and use a couple of minutes to contemplate over which type of statistical numbers should we use in order to compare the different countries. Okay, the learning objectives of this short video is that you should be able to understand that ANOVA is based on separation of the total variance in the data. That is simply, the name is analysis of variance, and the analysis goes on that you separate the variances and compare them. That variation can have the form either systematic or random, and further that the statistical inference is based on the relation between exactly these two, the systematic and the random variation. This is called an F-test. Further, you should be able to compare the responses between different treatments, and the treatments here are, for instance, the 4 different countries or 3 different diets, and you should be able to do that by these 4 steps: model specification, hypothesis formulation, a test, and then a model validation. In this we won't go through the model validation part. And then you should understand that one-way ANOVA is a direct extension of a 2-sample t-test to more than 2 groups. So here goes. One-way ANOVA is this setup again where we have 4 different countries, and we say we have these 2 types of variations. We have the systematic, which is between the countries, and we call it systematic because we know why there are differences in the numbers here and the numbers here, especially on the mean level, and that is simply due to them being from different countries. So we know why. That is why we call it systematic. And then we have a part which is within each country, so the difference between these points within one country, visualized by the red arrow here. And this one we call random, and that is because we don't know why. These are from the same country, these wines, and they give different results, and we pretty much don't know why. It's just a high value wine and a low value wine in terms of ethyl acetate. So this is the random variation within each country. So we have a term between, comparing the means, visualized by the green arrow, and within, visualized by the red arrow. So here is an example of 2 scenarios. So the question here is, where are the differences most pronounced, and how can we quantify it? So we have the 2 scenarios A and B, and A up here is an example where we have some differences between the means. You can see that especially the mean of group 4 over here is lower than the means of the 3 other groups. And down here we also have some differences between the means. Again, group 4 is lower, group 2 is the second lowest, and so forth. But in scenario A, we see that we also have a huge internal, within each group, random variation. That is that there is a spread in the points around the mean, which is not the case for scenario B. So we would say that the differences between the groups are most pronounced here, basically because we have a lower within group variation compared to up here. So how to quantify this? Well, we make a split of the variation and say, well, how much of the variation is due to the different groups, and how much of the variation is due to the difference between each point and the center in each group. So here goes for the model specification. So here is an example where we say we have k groups. x11 is the first sample in the first group, x21 is the first sample in the second group, and xk1 is the first sample in the kth group. And then it goes up to x1n1, so that is the last sample in the first group, last sample in the second group, and so forth. You can see here that n1 is not necessarily the same as n2, so we don't need to have the same number of samples within each group. And then we say these data here are normally distributed around the center, which is specific for the group, and some variation. And we see that the center is specific for the group, whereas the variation is not. It's the same variation across all the groups. So this is one way of formulating a model for one-way ANOVA. This is another where we say we account for each observation ij being equal to a group mean, mu I, and then plus some random variation. And then we specify that this random term, Eij, belongs to one normal distribution centered around 0 and with a variance term. And then we state that these ones are independent, and we say that this covers all the samples within each group for all groups. I deems the groups 1 to k, just as down here, and j is the number in the groups 1, 2, 3 up to n1, n2 and so forth, accounting for all the data. These two models are exactly the same. They're written up differently, but they are exactly the same. If we want to use these models to compare the means within each group, we can state a hypothesis. And the null hypothesis is that the mean for the groups, my 1, 2, 3 and up to k, is the same. So that is our general purpose by doing t-test, is to compare the means just with two groups, and that is the same purpose with one-way ANOVA, is to compare the means just with more than two groups. And here we want to compare the means of k groups, and we do that by assuming that they come from the same distribution, which means that they have the same mean. And then the alternative hypothesis is that at least one group is off. That is, that one of these equal sign does not hold, or one or more than these equal sign doesn't hold. We do this by splitting the variance, and we keep track of the variances in such a table here. We call that an ANOVA table. So we say, well, we have a total variance. The total variance is the spread around the grand mean for all the observations. So you see that this term is familiar when you calculate variances. So you take each observation and subtract the grand mean, which with double bar up here is the grand mean, and square them and sum them. That is the total sum of squares. And this number is your total variation. And this you split out on the two ingredients that you have in your model. You have a factor, systematic variation, and a residual, which is the random variation. So you keep account of your total variation. And then you keep account of the number of degrees of freedom, and that is the total degrees of freedom is n minus 1, because you calculate the grand mean. And this you split out on how many is accounted for by factor A and how much is left for the residuals. So these two numbers also sum to the total. These two numbers sum to the total. So we keep account of the variances and we keep account of the degrees of freedom. Then we calculate some statistics. we calculate the mean square of factor A, and that is simply this number divided by this number, and the same goes for the residuals. These numbers we use in what we call an F-test, where we compare the mean squared of factor A with the mean squares of the residuals. So this is two variance terms, and we divide those. And then we translate this F-test into a p-value. So the estimate here for the residual, the MSE, is actually the estimate of the variance from the model. And that is really nice to know, that this term is estimated by simply calculating the residual sum of squares divided by how many degrees of freedom is used to calculate it, and then you get MSE, which is a variance term. The test of the F value. So we could say, well, the F value here is a ratio between the mean squares of factor A, and this one is high when there's huge differences between the means between the groups. And this one is low when the means between the groups are fairly equal. And then we divide by the residual sum of squares, and then we get an F value. So intuitively we would say, well, if F is high, it gives confidence on that the null hypothesis is not true. Whereas if F is low, it means that the differences between the means are much lower than the overall residual variation. So what we would like to test is the possibility of getting what we observe and something which is more extreme compared to the null hypothesis. And that is the area here. So if we observe an F value of 2, then we would like to calculate the area up here, and that is our p-value. So this was shortly on how to formulate a model, state a hypothesis, and how to read off a variance table. In total, we will get the ANOVA cookbook, which goes like this. First of all, you should always visualize your data. Then you should consider whether you need to transform the data prior to any analysis. Then when you have done these two things, you specify a model and some relevant hypothesis. You calculate the ANOVA and maybe adjust the model, which means that you remove terms, and you check the model assumptions. In this lecture we will not go through how to check the model assumptions. That is a subject for a later lecture. Then you compare the contrast with confidence intervals, or a test between the differences of pairwise of means. We'll not go through this part either in this lecture. And then you comment on the results. So let's see how this is done in R. So here I have some data. So this is 40 samples, and they come from a study where some mice have been fed 3 different diets. So there are 3 different diets here. There is 2 high-fat diets, one which is combined with casein, that is milk protein, and one which is combined with whey, that is another milk protein. And then there's a low-fat diet where it's combined with casein as protein source. And you see here that there is 15 in the 2 first group and then 10 in the last group. The first part is to visualize the data. So we do that by saying we want this factor fat protein, and what we would like to see is the weight at the end of the trial. So this is the points. Let's put it on a box plot. And let's add on the individual points. In this case we have 40 observations, so it's not a problem to put in the points. There is not overwhelmingly amounts of data, so it's nice to see the original data. And so what we get here is the different diets on the x-axis and the weight of the animal at the end of the trial, that is after 14 weeks. So we could consider transforming these data because it seems like that we have a higher spread in the upper group, in the group with the highest response compared to the group with the lowest response. So in the model we specify that this should not be the case, that the variance should be similar. So let's just specify the model here. So the observations here, Xij is the weight at week 14 after the trial, and Xij is equals to μi plus Eij, where μi is from this group, low fat casein, high fat casein, and high fat whey. And J is equal to 1 up till Ni. And then we say that Ei is normally distributed with zero mean and the same variance across the group, and then this one is independent. And when we talk about transformation of data, we focus on that this guy here, the variance is the same regardless of which group the residuals come from. And if we take a look here, it seems like that the variance in the upper group here is higher than the variance in the lower group. So we might want to transform our data. So let's just try to put in a log transform on this guy. It doesn't really remove all the problems. We still see that we have slightly higher variance in the upper group compared to the lower group, but it removes some of the problems. So for now we just stick with this transformation. Then we make a model, and that is an ANOVA model. Use these data. This is our response variable. And then we say, well, the response variable is a function, that is this sign, of the factor fat protein. And here we only have one factor because it's a one-way ANOVA. So this is our model. And if you write it out, you'll get some sum of squares for the two parts. The factor, fat protein, and the residuals. So the hypothesis that we want to test is that the means of the three different groups are the same, so that the mean after, which is the weight at end of trial after 14 weeks of the diets, of the different diets, are the same. So based on the plot here, it seems like that is not the case, right? It seems like getting high fat diet combined with casein boost the body weight at 14 weeks compared to the other groups. But it can be hard to tell whether these two are actually different. So the model that we have built, we can use to analyze the factor fat protein. So what we get here is the ANOVA table. We do not get the totals, that is what we sum for the degrees of freedom and the sum of squares, but we get the partition of the sum of squares into the systematic factor, which is 0.4, and the residual, which is also 0.4. Then the calculation of the mean squares and the f-value, and the f-value here is 17 or 18, and we would like to know if 18 is a large number, and we do that by calculating a test, which tells us that it's pretty large. So in conclusion, we would say that it seems very unlikely that the three groups come from the same distribution, that is, that there is no effect of diet. It seems like there is some effect of diet, while we must reject our null hypothesis and accept the alternative, which is at least one is off. From this analysis, we don't know which group is off. It seems like it might be group with high fat and casein, which produces higher values, but are there any difference between these two groups as well? We don't know. So this is a subject for another lecture later on. Thank you.